Can everyone hear me? Excellent. So thank you for inviting me here. I'm really excited to be traveling again to do my Duckweed promotion. As some of you students have heard today, I'm totally in, right? So what I hope to do is to infect you with kind of my enthusiasm, okay? And that if I could do that, I accomplished something, right? So, but I will try to emphasize some points about the science behind this whole platform of new cropping. And I hope you will enjoy that part just as much as the commercial end. But first, let me tell you what drives me, right, in my stage, at this late stage of my career, to start something new. And this is like about maybe 12, 13 years ago. I start to realize that the world is facing a lot of big problems. And this is summarized here by Martin Blase's nice article in MBio that talk about the challenges that this planet is going to be facing, which is water and food. And sometimes we forget, right, what are the most important things in life. We get all angry sometimes every day, but actually, if you don't have food or water, you will really be very angry, okay? Never mind gasoline or, you don't have bread for several days. You won't want to know how you would feel, okay? Because gasoline is the last thing on your mind. So if you think about the way the world is going right now, there's a problem, right? There's like 9.5 billion people predicted. We are on track to achieve that by 2050. That's not so far away, right? It's like about 25 years from now. So think about that. Some of the younger ones in this audience, you're going to be facing that challenge because we estimate you will need about 60% more of increase of our crops that we are familiar with, which is maize, rice, wheat, and soybean, right? Those are the top big ones that most of the world's population are dependent on. And you need about 60% improvement in those. And you will think that this is already very big, but I want to convince you we have something bigger that you can do, okay, right? More than 60%, okay, right? But the other thing that you realize, and you heard about all the extreme drought this year, water is actually just as scarce and will be more scarce. If you live out in California, you'll know what I mean, right? Because the Colorado River is drying up. Can you imagine that? People used to go whitewater rafting. I don't think the water is that white anymore, right? So something to worry about, and this is real, right? Even I think oil companies are not denying that anymore, right? That there is actually something going on, right? Surprise, surprise. What I want you to see is really, this is for people who have been living under a rock for a while and didn't see this data, but I want to reiterate that. If you look back at the ice cores, 800,000 years, yes, there are fluctuations in carbon dioxide concentration in the atmospheric content, but it's never been like this. You see this shot up? It's scary, right? Because we are living in an experiment right now. You and I are lab rats. You may not know that, but you are in the biggest experiment of all time on this planet. This never happened before in recent time that fast, right? We are talking about less than 100 to 200 years. It went from this, we passed 420. Actually, this is an old slide. Last year, we had the distinction of passing that mark. So this is no joke, right? And you look at what is the evidence? People always say, well, the evidence is in the ocean temperature. There's a lot of water in those oceans out there. So as you may know, that water actually doesn't change all that much in that temperature. Guess why all the coral is dying? Well, look at this trend, right? It might not seem like a lot to you, 1 degree difference. Well, for them, that's a lot. And the cyanobacteria and other things that's living in those coral are saying bye-bye, because it's too hot for them. What happened, right? This is pictures taken this year. You see the effect of climate change in the developed world and the underdeveloped world. This is serious. And people are recognizing that. Well, the polar bear knew that long time ago, but they are almost gone, right? Because all the ice in the Arctics are gone in the summer. That's never happened for a long, long time before, okay? So I don't need to be a doomsayer for this audience, but I'm just wanting you to know that if you remember the previous slide before that one, we're supposed to increase our crops by 60%. How are you going to do that in the presence of climate change? Because what we are forecasting is that climate change will reduce corn and soybean yield by 40% by the end of this century. So we're going the other way if nothing else happens, right? And environmental problem. Our present-day agriculture is contributing to the greenhouse gas. 11% of the greenhouse gas from this country is coming from agriculture. And that's not just from the fat of the cows, okay? That's from traditional mechanized agriculture, right? And of course, using fertilizer and chemicals that you spray on your land and your crops, right? Fungicide is amazingly poisonous. We spray a lot of that on our crops, like potatoes and so on, okay? Trust me. And we need that to maintain that high productivity. But at the same time, most of those things are affecting the biodiversity in our soil and in our environment. So you have this tremendous also need of having fresh water to irrigate your crops. So if you're in California, you have to grow your lettuce, but your neighbor needs to drink. Well, I think my lettuce is more important because I have to feed my kids, right? I can't argue with that, but soon we might have to decide whether to feed our crops or to feed our family. That's not that far from now, right? So also, health, right? And also famine. These are things that, our crop is facing challenges to provide nutritious crop that are increasing productivity, and that's difficult. And of course, waste. Currently, most of our agriculture is centralized, which means that a lot of our crops are grown in big, corporate farms elsewhere, like in the Midwest, actually, right? And California. And you have to truck all that produce across country, thousands of miles in refrigerated trucks. Just think about the kind of madness, right? The energy that's involved in shipping those things to your, your fork from the farm, right? So the addition is that we're almost going to always demand more arable land because of the growing population worldwide. And that, a lot of the food is lost even before they get to your supermarket because of the perishable nature of this transport. So the point is, I'm trying to convince you is that we need new crops. Yes, we need to improve our current agriculture, right? To improve its sustainability and productivity by whatever means possible. You guys all know that better than I do. But what I want to tell you is that I think we also need new crops and new methods of producing crops that can meet these urgent challenges, and they can augment what we have. We need to look for additional solutions. So I always like to say, well, I don't know if any of you watch I Dream of Jeannie. I'm showing my age, right? But but you find a bottle on the beach like Major Hilly, and you try to like rub it, and you say, well, the genie came out, and you say, I'll give you an ideal plan, right? What would you want? What are the qualities of that ideal crop, right? That you love to have, but it's not there yet. So, maybe the genie can make for you. I was saying scalable is important, because that's one thing that will allow you to have delocalized or local farming, right? That will actually be economical and cut down on all the energy and greenhouse gas footprint. To get it to your table, within a 10-mile radius would be nice. Okay, 20, okay, but not 200 or 2,000 miles, right? Climate resistant or resilient will be very, very good because we know climate change is, we're not going to turn back. We are talking about minimizing it to go to 5 degrees, right? So, so something that you can really protect it from being, destroyed by the oncoming hurricane or drought will be good. Environmental friendly. You don't want something that, you would have to waste a lot of water or have to use a lot of pesticides or herbicides to grow. You want something that really allow you to grow it in a friendly way, right? And of course, you want it to grow fast. Who can wait for the plant to grow, right? We need food now. Definitely, you want highly nutritious plants will be very good because you don't want to just feed the consumer with calories. You want to feed them with good proteins and vitamins, right? I'm very, very basic, okay? I'm a pragmatic person. I'm trying to talk about famine and death, okay? So flavor will be the last thing on your mind when you are hungry, trust me. But it will be good, it will be good. So we will work that in after we get there, right? And finally, we want something that actually are easy to process because if it takes tremendous work and cost to harvest it, it's a no starter. So you want something that's efficient and economic for harvesting your product and converting it to a bioproduct, right? So that you can go to market easily and cheaply. Then you have a chance to be successful. And should I just sit and shake my head, right? See, I still remember what Jeannie does. Duckweed. That's what the genie would have given you, right? Not the potato buds that Matt Damon wrote in Mars, because it will take too hard. Well, if you watch the movie, you know what I mean. It's kind of gross too. Duckweed, growing duckweed will be much less problematic, trust me, okay? Because as you'll see, okay, and I give you some example at the end, hopefully I get there. But what is duckweed? Many of you have seen it, but you probably don't know all these scientific details. Yes, I might turn some of you off, but bear with me. We are in a science talk here, so I do supposed to tell you science, right? So it is a flowering plant, although most of them are asexually. They prefer to just grow. So they forego sex and they just want to grow and compete with the algae and cover the pond. That's their mission in life, and that is good. Trust me, it's good for humanity. But it's a flowering plant, just like your orchids or your Arabidopsids, if those are your favorites. It's in the family called Lemnaceae, and there are 36 known species. They are the smallest flowering plants, and obviously they range from size that are very small, from less than 1 to 20 millimeters, okay? These are some of the examples shown here. This is the largest one called Spirodella pyricer, or the giant duckweed. These are the common ones called Lemna minor, or the common duckweed. That's their common name. And these are the wolfear, the smallest. And you'll hear a lot more about wolfear today, and you'll get to know it inside and out, okay? I promise. So these are, now you know what they are, what they look like. They are monocots. They are aquatic, and they basically float, right? This is a key characteristic you have to remember, because not so many things are like that. They are truly floating plants. In other words, they're not like, water hyacinth, which have these roots that go down into the mangroves area, and just, you couldn't get rid of them. These are floating plants. They are relaxed, just like you're in the Bahamas and you're with your Mai Tai and you're just floating on the swimming pool. Yeah, that's it. So that's what they do. They are very just relaxed little plants, right? So they're easy to harvest. You see, this is the trick, right? And they are perennial because they just keep growing. This is a plant that just keep on giving. They keep dividing, making more and more themselves. And if there's no waves, they just cover the whole pond, right? And They are distributed throughout the world except in Antarctica. Although was there once and I tried to look for it, but I was in the kind of spring, not the summer, okay, of Antarctica. So I will go back to look harder. So I'm not giving up on Antarctica yet, okay? But except in the desert, right? I mean, definitely you won't find this in the Gobi Desert, unless it's really close to some, ponds that, oasis, and they brought it in, okay, manually. But lastly, they have strains with high protein or starch. And the magic is that they naturally have this low lignin level. So they're easily digested. You don't need to engineer them to have low lignin. They already come with less than 5%, okay? So this is why you will not get indigestion. You can feed it to fish, chicken, cows, whatever, or me. I actually eat them raw, right? Like a nice microgreen. So this is kind of my slide for you to get familiar with duckweed. And they are the fastest plant biomass growing, kind of producing plants. And for example, if you take this one of the smallest of the duckweed called microscopica, Wolfia microscopica, and you give it everything that it ever can consume. So it divides every 30 hours, and you can work out the exponential math. And if no fowl come to eat it, after about 4 months, you can grow to 10 to the 30 Wolfia, and that's about roughly the volume of this planet, okay? Even though it's only 1 millimeter wide, okay? Because that's a big number, 10 to the 30. And the potential biomass yield is about 4 metric ton per day per hectare. Not too shabby, okay? This is fresh weight. And in terms of dry mass, you can get up to about 100 dry ton per hectare per year. This is just from the ponds, okay, right? One layer. So this is already more than what you get from corn. And the one trial that has been done 30 years ago in Bangladesh showed that you can achieve easily about half of what potentially is the max, okay? So it can be, it's known, if you just go to any wastewater area, naturally duckweed just populate it because it's there, okay? And there's no, kind of deterrent from it. So anything that it has a nitrogen phosphate and in the right condition, they will be able to populate it. So the lack or what is missing is that there is no domestication, systematic domestication of duckweed as a sustainable crop. And why is that a problem? Because it's different from crops that have been grown by people for 10,000 years. It's not the same, right? This is something that's grown on water, not soil. It has its advantages, but you need to work out the kinks, right? To make it grow. And as I'll show you today, you also need a consumer base, right? Your base that wants that stuff, right? So you need to create a desire, okay? So Michael Pollan, some of you might have heard, right? He's a nice writer, really great writer. And he has a book called Botany of Desire. I suggest you read that. Because what it talks about is some great example of how humans choose which plants to domesticate, right? And it's not just a cultural consideration. A lot of it is economic, as well as, just happenstance, when you get to it, right? And it's really interesting because humans have to make choices all the time. And so you can choose duckweed or you can choose something else, but I hope you choose duckweed. Right? But so what is the challenge? What you need to develop a whole new crop system, right? I imagine, right? So the first thing is you would need to have a germplasm. Just like our ancestor 10,000 years ago, you start collecting seeds and try different ones, right? To see what has the right property that you desire, right? That's Michael Pollan's book, right? You really are driven by desire, what you do, right? So how it tastes, whether it's psychedelic, right? Okay, I mean, that's how don't laugh, right? Weed is right now become, not duckweed, okay, the other weed. It's actually big business. Why? Desire, right? There is something there that the plant makes that you want. That's why corn has been bred to have sugar. You want sugar, they'll give it to you, right? So you have to kind of find out what is in this plant that makes you want it, and then how to grow it, right? So the base is to start with the germplasm, optimize it, right, with target products and market gene to breed it, right? And to have to find the right traits that makes it desirable. And then you have to do all these other things, right, to actually make that conversion possible. You have to learn how to produce it. So this is what we are almost finishing, right? After, gosh, about 6-7 years, okay? I tell you this, especially for the students. So nothing happens overnight, especially something that you believe is new and is important. Keep in mind, you're going to be in the long haul, right? You got to keep that vision and keep that, ambition, as well as believing in yourself, right? To make it real takes time. But this is what I will talk to you about, what we're going to be doing. We have started to do some parts, but all these other things have not been done so much, okay? So it's really a partnership between the field work and the lab, right? Because I go and collect duckweed all the time, okay? And it's all over the world, so you can easily do that. So today I'm going to give you some parts of areas of service, research, and development that I do. And I have to do this. Because, because if you don't do this, or at least encourage your friends to do it, which is even better if I could, right? Right, Mike? So if I can encourage others to do what I want to see, I don't need to do it, then even better. I think so I think these are the parts that I will tell you. So I will quickly tell you the, the germplasm collection that we have started from 2009. So that's 13 years ago, okay, right? And it's now, I think, start to see results, right? To see that it actually help people to get access. It's not for us. I mean, it's really for the community, right? So we started back in 2009, as I say, and I'm not going to read all that. So currently we have 900 or so sessions with representative for all 5 genera. As you see, this is how many different types of duckweed there are. And our mission, I want to point out to you, is really to facilitate and broaden the use of duckweed through the community sharing, right? And characterization of the natural diversity of the genome and phenome level, because you need to have this base to understand what you can do and impact. So we set up high-resolution genotyping strategies to characterize and extend this resource, because without the ability to tell the duckweed apart, which is not as easy as you might think, or if you don't think, but I can tell you it's not easy because they're small and they don't have much architecture, right? They all look the same, but they're not, okay? And that's a good thing. Here they are, the mugshot. So I actually started not only the international conference, I also started these the steering committee, okay, international steering committee. And part of the responsibility of our steering committee is to create a quarterly newsletter, which talk about all things duckweed, right? If you're interested, email me and I send you, put you on our mailing list, as well as to send you the latest issue, so you can get up to speed, right? What has been published in the duckweed field in the past 3 months, and other commentaries, as well as student spotlights and sharing of announcements and so on. So it makes you feel like there's somebody out there working on duckweed too, right? So you're not so alone, right? Oh, I'm so weird working with this little plant that nobody cares about. No, that's actually growing population of people who are duckweed people, okay? So you won't be alone. But I think one of the things we did was to actually demonstrate that there, in fact, is a lot of trait variance, which is good because that tells you that there are a lot of natural variation and potentially useful traits, right? And I'll give you an illustration today, how that works out. So give you an example. We did a preliminary screen of 50 strains from 5 different species that are globally distributed. And what we find is that the growth rate can vary by 6-fold between different strains. And starch content can vary by like 8-fold. So that's a lot, right? If you have a maize that's like 8 times more starch or sugar, you know, that you can go to patent it, right? And total protein level that can be changed by 4-fold, right? Again, it's like between 10 or 40%, huge, right? Well, that's what you have nature for, to make variation for you. You don't need to engineer it. So now, once you have this germplasm, what do you do with it? Well, one of the first things we know to make a competitive, right, field to attract good biologists like you and you, right, into it is to create the resource, right? To enable people to do cutting edge science. Because if I'm a good scientist and I don't care about making money, but I want to learn things quick and make my mark, well, you need a good genome. I think any good card-carrying biologist would like to have a good genome that they work with because it will allow them to use a lot more tools than otherwise. So this is what we spent the last, I would say, 8 years trying to sequence, well, me meaning my friends and I, okay? And I'll acknowledge them later, so hopefully bear with me. But I just want you to be excited to give you a little bit of lesson in the phylogeny and diversity of duckweed. They're in the Araceae family of monocots and in the Mateli, together with Sostra, which is the seagrass. That's its closest relative in these two families. Their closest relative, other branches will be like asparagus and and other kind of monocots, right? But Poales, where you have all the seed crops and things that you are used to around here, actually a little bit much further away. So they are at the basal monocot position. So evolution, I was talking to Mike, this is actually a very interesting kind of junction to try to, if you want to understand how enzymes and genomes evolve and how does, speciation occur. This is a really good family to have as a comparative analysis because it tells you or shows you how things can change from very early on after the early progenitor split between monocot and dicot, right? Eudicots. And of course, the cool thing is that this is Wolfia in the jar, right? You can have like 1,000 of them in a little test tube. How good is that, right? You can do population biology in, Eppendorf, right? Amazing. But then you also have all these variety. I love this one. This reminds me of the Klingon, weapon, right? That they throw around. So duckweed has this remarkable ability also to change and adapt. And you can see this, kind of pictures that illustrate that. Now, in terms of genome update, this is why I want to get you excited. We actually now sequence many, or have sequenced it, although they're not published as some of them, but they are available, all the finished high quality reference genome. You can see all these green stars are actually distributed across all 5 genera, okay? This is this is taro as an outgroup, okay? All these are the 36 species of duckweed. And you can find this in our review article in Plant Cell last year, okay? So you don't need to quickly copy this down. But what I was not in there is to show you this updated sequencing. So the thing I want you to get excited is that you see all these 5 genus genera are now have representative genomes, okay, in hand, that we can really start to understand how the different things diverge and so on. So I think it's a very exciting time from the first publication of a 20x reference genome of Spirillella back in 2014. We are talking about 8 years, and we now finish, I would say, almost 10 genomes. I think that's pretty good, okay, of 10 species with multiple representatives in each one. I always believe in twos, okay? Not that they will cross with each other. I'm not Noah, okay? but it's that when you make genome assembly here, if you have two independent strains assemble simultaneously in parallel, and then you compare them, it gives you much stronger evidence for, your assembly being correct, right? So this is the lessons we learned. But what you see is a tremendous variation from 150 megabase all the way to more than 2 gigabase size. So this suggested to us, and it's being shown now, that there are polyploidization as well as hybridization, okay, going on in the family. And this is part of some of the exciting things to ferret out all these things and really try to create good model system to look at epigenetics as well as how polyploidy actually changes the complex of the what genes are lost when you have this ability to lose things and not get weeded out right away. So I think it's an exciting time, and the genome sequencing technology really helped. Just to show you that, in fact, some of the interesting thing we learned already from the Wolffiella genome, we compare it to the Spirillella, which is the first one, right? That's the basal one. And the Wolffia genome. Wolffiella and Wolffia are the two rootless duckweeds. They have evolved away. They are mutants, right? You can think of it that way. And they lost roots completely. And they lose, also in Wolffia's case, vasculature, right? There's no more xylomal phloem. I'll show you the cross-section, so you'll get to know it inside and out, as I promised. But Wolffiella, we haven't done so much cell biology yet, but it's different. And what you can see is that here we curated most of the disease-resistant related genes to show you that every of these plants, they do things a little differently. I just want you to see, one of the first things that, and I'll talk about a little bit more later, is that Wolffia actually removed a lot of these so-called NBR genes, right? These are the disease-resistant locus, the R gene that you learn in classroom, where in most dicots and crop plants are highly amplified because they are supposed to be the immune providing receptors, right? Genes that you learn about in the classroom. Well, Wolffia actually don't need them. So they have only 3 or 4 of them left. So interestingly, Wolffiella, another rootless duckweed, actually didn't like it that way. They actually, increase rather than decrease compared to Spirillella, right? So just to give you an example, for Wolffia, instead of the N genes, or R genes, sorry, what they amplify or what they prefer to have as maybe a defense mechanism is these, leucine-rich repeat receptor kinase like proteins, where they go up to about almost 100, where compared to Spirillella and Wolffiella, they are around the 50 to 40 kind of region. Just want to give you some example how different plants open new windows of understanding new biology, right? How does Wolffia become resistant without our genes? That's probably something you didn't read in any books yet, but someday we will learn, right? How that actually does that. So that's just to tell you that duckweed could be very powerful basic research tool as well to understand the diversity of ways that nature can use to perform the same function, but in a different context, okay? So now I want to give you a drill down, right, really deep into one story, because people that talk to me today know I like to talk, because I like to tell stories, but I will just tell you one story, okay, so bear with me. So this will be the story that's published last year with the Wolffia genome, where when Todd Michael and I decided to sequence it, he said, well, what should we do about this? I mean, you can't just have a genome. We had to do some biology. And I told him that he and I both work on circadian, right, control. He worked on it a lot more than I did, okay, I have to say. So he's a guru in circadian control. So he immediately agreed. We should look at the circadian cycle or diurnal cycling of genes. And the reason is because what is interesting about Wolffia, as I remind you that I just told you that Wolffia has the species that grows the fastest. So we would like to understand how this little plant manages genes to enable this fast growth nature of doubling every 2 days, which is remarkable, right? So, okay, so he agreed, and I'll tell you the story, what we learned. So remind you, Wolffia is phylogenetically the most derived because if you compare the barcode sequence from the plastidic barcode, compared to Spirodella is the furthest away, okay? And Lemna and Wolffiella are here, and Landotia represented by a single species. So this is introducing how duckweed grows. And as I try to emphasize here, it's a plant built for continuous clonal propagation. So this thing likes to shoot out more babies. Okay, so literally in the same plant, you will see the mother frond, which is here, the first daughter frond, which is here, the second daughter frond, which already made and is starting to mate its own daughter. Okay, and of course the granddaughter, first granddaughter is here. Okay, this little thing is called the stipe and is contiguous with the meristematic region, which we are trying to understand. It's not your typical L1, L2, L3 layer, okay? I can tell you that. But it's a very intriguing thing. So now you know duckweed from the inside, and this is duckweed from the outside, right? Wolffia. This is the dorsal part. That's the top facing the air, and usually it's out of the air. And this is your ventral part, which is a lot of the cortical cells, and it have this keel-like structure for this species that keeps it righted, right? If you're a sailor, you know what I mean, right? That's the convergent design, right? There's no architect there, okay? You don't need it. Nature is the architect. And it's a beautiful picture that my collaborator made. Okay, I didn't make that. It's from my collaborator that I'll mention to you later. So now, what we know is, as I mentioned, it doubles every 2 days. What we want to do is to reveal what's the strategy, right, for this fast growth in this genus. And what we also want to compare this genome to the basal Spirodella, the most complex of the duckweed, to see what had been removed and what has been gained, right, in terms of genes. And so we suspect that because all organism actually has a clock. I hope you all know that, okay? And why? Because it's a selective advantage to predict and anticipate what's going to change in the environment. So you do the right thing before it happens, okay? Unlike what we know climate change is happening, but we just don't want to do anything, right? That's very different from nature. In nature, we predict something is going to happen, we do something first, so that when it happens, we're right there. That's why from bacteria to man, we all have a circadian clock in some way to kind of have predictive behavior internally and externally, right? To maximize your competitiveness, right? To gain resource. So what we wanted, suspected in, this is the beginning of our project, is that we suspect Wolffia may have evolved a more advanced network of coordinated gene regulation. Right? This is what you might think. If I want to control this to death so that I can be optimal, I should make more genes, right, in the clock and control more things so that they are coordinated and optimize the coordination. That's what human would do, right? That's what Washington do. Come on, right? They never fire anybody. Nature does a different thing. This is spoiler alert, okay? sorry, but first, after we sequence the genome, we found out that wolffia actually, this is for Mike, it emphasized even more sphingolipid related genes. So first off, if you look at Spirodella as well as the wolffia genome, 2 of each, we find that there are a bunch of ortho group. These are gene families that are in the sphingolipid wax biosynthesis, ammonium ion uptake, and so on, and these endopeptidase for potentially resistance, okay, and other type of processing. However, in the specifically suppressed or underrepresented genes that wolffia got rid of, what you see is that they are in these flavonoid pathways, cell wall biosynthesis, terpene biosynthesis, and immune response, just like what I just showed you, right? All these known immune-related genes are removed. What they are emphasizing, and I told Mike that this is very interesting, to look at lipid-related stuff, especially sphingolipids, which are well known to be involved in signaling, okay, in biology. I hope you guys know that Mike's a great guide here to talk to you about that, right? And then you have these other genes, again, even more amplified than in Spirodella, right? So suggesting these are the places we should look to see what these pathways are doing in wolffia to help it grow fast, right? This just to show you that the genes are remarkably synthetic. So if you find these genes in the order in Spirodella, Pyrrhiza, you find them also on the same chromosome counting in the 2 strains of wolffia that we sequence. Just to show you that we can track, right, how genes evolve. The reason because we want to know, why is it rootless? How did it become rootless? There's a gene called WOX5. This is related to Wushou, which is the key, WOX5 is a key homolog of Wushou that specify the stem cells of roots. Lo and behold, wolffia doesn't have this gene. The question is, how did it lose it? Well, because it's synthetic, we can look for its neighbors, right? And then go back to look in the wolffia genome. And what we see is in the place where you expect a wolffia gene, all you find is transposable elements, right? And we see that in several other genes of similar nature. This is the NPH3 gene. This is the WOX5 gene. So you see the same type of, both genes are knocked out, right? So I think that these are the take-home lessons, where, genes such as those for root specification and light-dependent growth have been lost, as you would expect. And then T insertion and proliferation seems to be a mechanism for this attrition. And finally, one of the take-home lessons is that the loss of these NR genes that are highly conserved and proliferating in other terrestrial plants, the other plants, suggests that wolffia do it differently. And it will be remarkably interesting to learn how it does it without the N genes, okay, right? All this basically to provide maybe more economy of resource. So this is why when I discussed it with Todd, I said, this looks like it's trying to skim on things, right? So, so this is where there's transcriptome, right? To look at actually how many genes are really regulated in wolffia by the diurnal cycle. So we did this 12-hour light, 12-hour dark cycle and collected 13 time points. So I had to give credit to my student, right? Every 4 hours over 2 days, they have to take team, right, to collect samples. Very big job. And then prepare all these RNA. So somehow I get them to do this thing, crazy things, right? This is what you see. It's a beautiful picture where you assemble this picture, and this represented by 1,600 genes here. Every row is the same gene. It's color-coded, right? Basically, by the expression level. You're probably used to seeing this. And what you see is that the red is always the high. So all these genes are cycling in groups, right? Through the diurnal cycle, just as you expect. I think this is it's almost like art, right? But it's actual data, because what we can glean is then the genes that are actually known to be circadian regulated genes in the dawn and evening, you see them cycling exactly where we predicted, right? they are in the circadian dawn, they peak, and then the next phase comes up. And then for this group of genes, they do that, and then you have other type of phasing, right? Just to show you that it works. Now, what's cool is that this number, 1600 gene, is too little. Because in Arabidopsis, it's about 30% of the genes that's controlled by circadian or diurnal cycling. Here, we only found 13%. So basically, Wolfia is too lazy or too cheap to regulate. Because by removing regulating mechanism for these genes, perhaps Wolfia can really economize, and that's why it has such a small number of genes, 15,000, right? That you can actually remove a lot of things. So by doing this kind of Venn diagram, we can see what type of processes or pathways are actually kind of not regulated anymore, but is regulated in rice and Arabidopsis. Whereas these are the genes that actually Wolfia bothered to regulate that are actually not being regulated in these model plants, right? So the point is that the system choose what is important for them. And for Wolfia's case, it chooses the right mixture of things to minimize the extra thing that they don't need. And thereby, they are the minimalists, right? That's in now, right? Supposedly, like Elon Musk doesn't even have a house, right? I heard. I think he can afford one, but I think he what I heard is that he wanted to prepare to go live on Mars, right? So he live in a trailer or something, right? But the point is less is more, right? That's the take-home lesson that we learned from Wolfia, that there's a way, if you grow fast, you might not need to regulate a lot of things, and it's fine because you're dividing every 2 days. Maybe the pathogen couldn't keep up with you, right? There are interesting things that we still need to understand, but I think these are the tip of the iceberg that show us that there's interesting strategies of life that we don't know before, right? That is begging to be understood. All right, the final 10 minutes, I want to give you a rundown on taking up the challenge, right? So I told you about the exciting science that you can do and begin to think about, right? Having all these genomes, you know, being at a very interesting evolutionary position, and also having all these preliminary data that suggests they are doing things differently. So you're not rehashing what's been learned, right? I think that's exciting. But I think what's also important is to make sure what we do will become societally relevant, right? That we're going to make an impact in our lifetime so that it will actually be satisfactory in another way, rather than publication in Nature or Science, but actually seeing that it help people, I think will be, it sounded like grandstanding, but it's not. I think people really truly, I think, become very happy when they see what they do make a difference. And so today I want to tell you two things, and that's why there's two companies being made, right? One is to learn to farm duckweed effectively and efficiently. Because if you want to have a crop, you need that, right? You need to be able to day in and day out grow and produce what you expect and more, perhaps. And secondly, you want to develop strategy for product and market for this product, whatever you're growing. Because if you don't have that, there is no demand and there's no reason to do it, right? So it's a chicken and egg problem. But actually, chicken and egg problem, I have an answer. So who think the chicken come first in this audience? Sorry, I digress. But I actually talk to this with a lot of people, and it's an interest. Who thinks the chicken come first? And who thinks the egg come first? So you see, I have to say I go with the egg, right? Because the egg always gives rise to the same genetic makeup as the chicken that it makes. But the chicken doesn't make the same egg all the time. All eggs are different. So keep that in mind. That's a good lesson in genetics because it teaches you to understand, right? To see how deeply you understand inheritance. And not that you don't, appreciate your husband, but, I think, or spouse, I should say, because it takes two to tango. Anyway, so I digress. That's entertainment. It wakes some of you up if you're falling asleep. So the challenge one, grow duckweed at scale. You say, why don't you just go out and get a big pond, right? Like most people, or raceway. That's all fine and good, but it's hard to scale because it's expensive. You need to buy land and you won't be that climate resilient. Remember the magic plan, right? So you want to be climate resilient. You want to be able to grow this anywhere. So when the storm is coming, I put it back indoor, right? I think that will be great. So the approach and philosophy is that we want to utilize the unique growth habit of duckweed floating, right? Two-dimensional growth for vertical integrated growth system to maximize production per unit area. And then you want to modularize the system so when you have it complete, your kind of perfected your growth system, it can be immediately duplicated 10,000 times. Not that each time you have to go and dig in a new place and don't know what is the important parameter anymore, right? So I want to do it once and that's it. Then I can maximize everything based on that learning, right? And lastly, you want to be able to combine the sensor technology and automation so you decrease labor costs and facilitate the adaptability to truly precision farming using AI so you can optimize, right? And this is the vision, right? You can start small. This you can buy already from us, right? And we have worked it out. The next is to get it into many more tiers and automate it so that you can now truly scale to kind of vertical, just local. This is like for your neighborhood farm, right? That you get a factory and you just build it, 1,000 of these and you can do that, okay? And then you can grow truly to agricultural scale to about 1 million tons a day, right? If you have about 10 acres. So this is the numbers, right? At a top growth rate, about 20 grams dry weight per meter square per day. We have shown that this is the rate you can find from our strains, okay, under optimal conditions. So that's the potential growth rate, right? And this translates to about 130 grams of dry duckweed per meter square per day. And you work that number and it becomes 470 tons, right, per hectare per year. And this is 38 times what you get from corn today, right? And this is only from 6 tray system. It's scary, right? How much we can make in theory. Okay, so this is no longer 60%. I just want you to notice that number difference. Okay, we're talking about 38-fold, right? So give you more pictorial understanding. How much space do you need, Eric? This is a big football town, right? So let's take a football field. How many people can you feed? Well, a football field is slightly larger than 1 acre. So let's say it's 1 acre, right? If you grow it purely with corn, you basically can feed about 4 people. Estimated based on like calorie requirement per average size person. Okay, for me, maybe a little more. I eat a lot. If you grow soybean, because of the productivity is much less, you can feed maybe 1.3 people. Okay, that's hard to cut too thin slice. Okay, let's say 3 acre, you can feed 2 people. Okay, 4 people. Yeah, and then, but with 1 acre DPM system, I just show you, and increase it to like a 30-tier system, then now you can grow 2 million kilograms per year. Okay, right? And this will be enough to feed 2,000 people, right? Enough for a small town. We put Seaside Heights down here in New Jersey. Okay. Again, just to tell you, this is the potential, right, that we can reach. It's not, pie in the sky. How do we get people to know that way? Well, I talked to some students here. You got to let them grow it. That would be my passion, just to make all of you grow duckweed yourself, so you get rid of the like risk factor and like the unknown. It's so hard. Okay, strangers. So get duckweed to be your friend, right? So finding a niche or two. So the first niche of products is duckweed pods. So these are the turions that actually are the special structure that duckweed and other aquatic plants made so that they become dormant and sink to the bottom of lakes. So you can have duckweed in Siberia, okay, because they come back every spring. But they're remarkably stable. You just throw them in the refrigerator and you can have duckweed on demand. You can grow duckweed anytime by anybody. I mean, anybody. A 4-year-old can do this, okay? So we basically have this, you can order it for $15, I think. You know, it'll come with fertilizer, come with, you know, the duckweed pods. I call it pod because it's not an official name, right? It's escape pods. You watch Star Trek or Star Wars? Yeah, escape pods. You get the idea. This is how they escape and come back when the Klingons are away, right? So when the old man winter is away, then the duckweed comes back out. And this is what they look like. These are the little pods, right? They are dark in color because they're very rich in anthocyanin, very dense. And these are the fronds, okay, that mix them. And that's what we provide together with the little baggies of fertilizer so that you just need tap water and you can grow your thing. And then, of course, you can also buy fertilizer to keep on growing as you like, right? And the final slide is that additional food supplement that we have. And I talked to some student about starting some microgreen business, right? This is the urban fronds that my student Sean designed. This is fresh product that you can produce locally, right? And they have a long shelf life of 2 to 3 weeks in the refrigerator. And you just eat it like any other salad. And this is the magic powder that we discovered, Sean and I, that they actually made naturally the vitamin D that actually does all kinds of magic things in humans, right? Your bones, your skin, your hair, supposed to be good for many things. So these are things you can get now from, you know, our site, which I encourage you to go check out. And I like to, and of course, to acknowledge I didn't do most of the stuff I show you. I just encourage people to do them, okay? So, but the 2 people I really had to acknowledge in my lab who are current graduate students is Kenny Acosta and Sean. I don't have space enough to put the pictures there, but you know, Sean really they're spearheading all the commercial development with me. And Kenny had been with me since he was an undergraduate, and now he's almost finishing his PhD. And he has done a lot of the informatic work as well as the microbiome work that I didn't talk about at all, as well as many other projects he's involved with. So he's my, like, you know, well, left and right hand person. The people in blue are all the past, you know, students that I had and postdocs who had contributed, both graduate and undergraduate. I want to give a shout out to Ludmila Borisyuk. She's Ukrainian, and she did the nice cross-section and the 3D rendering of Wolfia, so that we now almost like the C. elegans of the flowering plant, where we have the knowledge of the number of cells in the whole plant, right? This is the plant where it's first done, okay? Because you can actually literally reconstruct the whole thing in 3 dimensions. And Rob Martinson and Todd have been compatriot in sequencing duckweed. All the genomes are done by them, not by me, although I help curate a lot of them. So I think they deserve all the credit in the genomic side. And John been wonderful to help us understand lipids, and he has a nice paper with Rob just came out, demonstrating you can make up to 10% oil in dry way in duckweed, okay? So it really opens up now the faith that it could one day provide the truly renewable biofuel continuously and just keep on giving, okay? So I think I will stop here, but before Vanessa kicked me out, you know, allow me. I told the student I was going to make a paraphrasing of Gandalf, okay, if you know Lord of the Rings. So please bear with me. So this is, again, you know, at the risk of sounding like a preacher, but I want to like just have a little fun, right? Because the corollary is remarkable. if you think about the mountain of Mordor and what's going to happen to this world and all the strife that's going on, it's not really that far. So as Gandalf would have said, as the Anthropocene, aka the age of man, now we should be thinking about the age of duckweed, right? Wreak havoc on the balance of our world and threaten the existence of life and all that we hold dear, right? There arises a little green plant, humble but unexpectedly resilient, like Frodo, right? And always can surprise you what it can do, right? Support it and nurture it, for it may yet catalyze the change. It won't be the change, but it will catalyze it to save the goodness that is worth fighting for in all of us. Thank you.